invited him to the seminar series at the Center of Personalized Medicine because not only he published this book, and the title is The Personalized Diet, and this book is certainly aligned with the interests of this center, but also because he has published seminal work demonstrating the importance of the microbiota in many different systems, living systems. Moreover, I'm inviting him to Oxford, not just because of what he does, but also because of whom he is. And I'll use a few minutes to talk about whom he is and about this disruptive journey so far. So this is publicly available, this slide in Google. Eran grew up in the southern Israeli city of Beersheba in the desert in Israel. And after completing elementary school, his family relocated to New York City, which was a tremendous culture shock for him, I would imagine. He started high school in Manhattan, not speaking a single word of English, not knowing anyone, nor being familiar with the New Yorker's 1980s culture. The experience of being an outsider taught him skills that he still carries with him until this day: willpower and the perseverance to overcome challenges. Following high school, he went back to his homeland Israel to serve in the army as a naval submariner for over 4 years underwater. And I'm sure that this experience further contributed to his persona. His curiosity for life sciences started in high school, and that same curiosity led him to medical school at the Hebrew University in Jerusalem, after which he was a gastroenterology resident at the Hadassah Medical Center. While he was a physician at the Tel Aviv Medical Center, he did his PhD in basic immunology at the Weizmann Institute of Science in the laboratory of Professor Archaea, who is well known for the development of chimeric antibody receptor T-cell treatment for cancer. In 2009, Eran went to Yale University to postdoc with Richard Flavell, focusing on the then and new exciting links between the gut microbiome and innate immunity. In his lab, he characterized the NLRP6 inflammasome and demonstrated that it played a role in host microbiome interactions and the risk of developing colitis, cardiac metabolic disease, and colorectal cancer. In 2012, he was recruited by the Weizmann Institute of Science to train the next generation of thinkers and to establish the first microbiome lab in the institute and also in the country. More recently, the DKFZ in Heidelberg invited him to direct the division of the microbiome and cancer. Personally, I first met him in 2018 when I had the privilege of giving a talk at the Cell Press Symposium Next Generation Immunology that he co-organized with Ido Ahmet at Weizmann. Last February, he organized another one of these meetings, and you'll be asking yourself why the subtitle of these meetings is Work Hard and Play Hard. And I think I know why, and this is why. Work hard and play hard. This was my experience of this meeting, and this was perhaps the most party-intensive meeting I've ever been, and scientifically also very intense, with excellent science, excellent food, excellent wine, excellent music, and excellent dance. Eran, I am certain that you will inspire the next generations in Oxford to work hard and play hard. And if it's not now, at least in the future, we hope, when this whole pandemic finishes. Welcome to Oxford, Eran.
Thank you very much, Anna, for a very, very kind introduction. And I'm thrilled to be speaking to this forum, although, you know, through the internet, given our current limitations. And I'll try to share my screen right now. Do you see it?
Yes.
Fantastic. So thank you again. It's really a pleasure. And in the next 40 or 45 minutes, I'm going to try and share with you some of our vision regarding host microbiome interactions and how we can integrate it into personalized medicine, which of course is the main theme of your great institute. And before starting, I thought of introducing four major concepts that have emerged in the last decade of the really young period of growth of the microbiome field, which I think are leading us towards the next decade of discoveries and will be important also in my talk. So the first concept regarding the microbiome is that we have a bacterial ecosystem or microbial ecosystem at every niche of our body, which interacts with the outside world. Of course, we've mainly focused on the gut microbiome, given the fact that it's accessible, it's complex and very important, but we increasingly are interested in other microbiomes and how they communicate with the host in health and in disease. And I'll give you at least one example of our recent work on this. The second concept relates to the fact that as we dove deep into the human microbiome, we found that it is highly diverse. And at first, it was very scary for us. We don't like diversity in our regular research, but when we sequence humans deeply enough, we find that we can identify different signatures of different humans. And we actually utilize this diversity in order to try and link the microbiome to different traits of the same genetic susceptibility of disease. And I'll try to give you an example of that as well. The third concept, which is very important in our own work, relates to the fact that we may regard the microbiome as a signaling hub that integrates into it signals that come from the human host. For example, the human host genetic system, the human immune system, impact the composition and function of the microbiome, but also a very large number of environmental factors, such as the one depicted in this cartoon, impact and signal to the microbiome. The microbiome integrates all of these signals, transmits them to the host, and we see our vision or mission in life to try and unravel the letters of the alphabet of the language by which we and our microbes communicate on a very intimate basis, which, as I'll try to convince you, is extremely important to every facet of our health. And the fourth concept, which has been increasingly becoming the center of our work in the last 2 years, is the discovery that microbiomes can live in one place in our body, but through their synthetic and modulatory activity, they can generate tens of thousands of small molecules, which we call metabolites, which can influx into the sterile host where they can impact very far away cells and organs. And with this, we can try to start to mechanistically explain how, for example, the gut microbiome may impact very distant organs such as the brain and others in health and in disease. And we take all of these concepts and we try to apply different technologies in order to try and contribute towards a better mechanistic understanding of host microbiome interactions. And if I break this down, we are trying to understand and learn how the microbiome impacts the physiology of humans in health in basically every process that we run into. We also are trying to understand how the human host impacts the composition and the function of the microbiome. And we try to understand what happens when the communication channels between the two partners of the holobiont are perturbed in a way that leads to susceptibility to acquiring common diseases. And with this knowledge, we increasingly believe that we may be able to contribute new principles leading to microbiome-mediated disease treatment. And today, I would like to share with you some vignettes of our work related to the treatment of person-specific microbiome treatments in different disorders. And when we start talking about microbiome-associated treatment, we need to tell the truth, which is that in 2020, we are still talking about three major utilized microbiome therapeutic avenues that are neither personalized, and in many cases, they are also not data or science driven to a sufficient extent. The first is called prebiotics, which is a dietary means of usually supplementing the diet with fibers in order to improve the health of the microbiome, whatever that means. I personally don't know what that means. A second method is called fecal microbiome transplantation. It's a brute force method by which we try to replace a person's microbiome when that microbiome contributes to disease with a healthy person's microbiome. It has been proven to be exceedingly effective in some bacterial infections in the hospital setting, such as C. difficile, colitis, but it's still very limited in more complex microbiome-associated diseases. And the third avenue is probiotics, which I will not get into because it's very emotional. But overall, I can say that the level of data supporting these treatment modalities is still highly controversial and limited in its conviction. But on the optimistic end, with the acquisition of huge amounts of person-specific data coming from the microbiome and its integration with other data sets that are acquired from the same individuals, with very advanced computational systems, we're increasingly able to utilize this system in order to understand disease traits and to actually modulate disease traits through the microbiome. And all of this is under development, and I'll show you some vignettes of our own work. And we can divide these research-based treatment avenues as 5 major avenues. And the first avenue is called personalized nutrition, and we've developed this in 2015, and it is kind of our science or data-driven nutrition-based microbiome therapeutic approach that is the correlate to prebiotics. Now, what is personalized nutrition? Personalized nutrition aims to utilize the microbiome in trying to optimize health. And so far, we've mainly focused on a group of diseases, which are, of course, not strange to any of us here in this talk, which constitute one of the biggest disease sets in our species history. They include obesity, type 2 diabetes, and all the complications, cardiovascular and others that accompany them. These diseases develop super rapidly in a few decades in front of our eyes. You can see on the top how they develop in the States. On the bottom, you can see the really alarming numbers, again, in the States of adults and of children recently that suffer from obesity and its complications. And the diseases that develop so rapidly cannot be simply explained just by genetics, which do not change that rapidly. So we're thinking environment. And when we think environment, again, the microbiome emerges as a potential hub that may contribute to these diseases. Now, when I started reading about nutrition and how it is formed to affect everything that we do in our daily lives when we try to come up with a quote unquote healthy diet, I was surprised to find that most of the data that forms the paradigm which we all intuitively follow comes from small scale studies from the 1970s, which basically are given different food components, numbers or grades. So for example, calories are is one such grading system which gives different food components different numbers. We try to mix and match these numbers in order to come with what we hope would be a healthy diet. An even more extensively used grading system for food is called the glycemic index. And take my word for it, almost any diet that you've ever tried is based in one way or another on this glycemic index. Now, what is a glycemic index? It's a system formed at the 1970s by very small scale experiments, which usually took 10 healthy human individuals who were given an identical food and their blood sugar levels were then measured for 2 hours. So for example, if you would take the 10 healthy individuals, give them an identical piece of celery, measure their blood sugar levels, you'll see in average a small spike, such as the black graph. This spike would be given a number, and this is the glycemic index of celery. Now, if you would ask the same 10 individuals to kindly eat an identical piece of chocolate cake, their average blood sugar rise would be higher, like the blue graph, and this would be transformed to higher numbers, and this higher number would be the glycemic index of chocolate cake. Now, if you would look now in your smartphone, you would find endless tables giving glycemic index to any food and food component on Earth. And take my word for it, any diet that you've ever tried, when you went to a dietitian, a physician, you bought a book at the airport, is based in one way or another on this glycemic index system. Now here comes the big problem. When we repeated this experiment, but not with 10 individuals, but rather with 1,000 healthy individuals, and gave them one of several identical foods. For example, on the left side, you can see these individuals when we gave them either 50 grams of glucose, bread and butter, fructose, you name it. And we measured their glycemic responses to these identical foods. We found that the average rise in the foods was exactly their glycemic index. So this was totally accurate. However, when we looked at the individuals' responses, we found a huge variability. So some people taking 50 grams of glucose spiked their glucose levels to a diabetic level. Others didn't care at all. And this was true for all test foods on the left panel that we've tested, and even more true for one of thousands of real-life foods that we've tested in this cohort on the right side. We've now completed close to 100,000 of these analyses on different people. And this gave rise to a very ambitious project, which we call the Personalized Nutrition Project, in which we took these 1,000 people and kindly asked them to give us a week of their life. And during this week, we've characterized their gut microbiome, we performed many blood tests, they filled many questionnaires, we measured them, we developed a smartphone app for this project, so they filled during a week of follow-up everything that they were doing, when they were waking up, when they were going to sleep, what they were eating, how much they were eating, and so on and so forth. And we've connected each and every one of them to a continuous glucose monitor of the type used on diabetics usually, which measures their blood sugar levels every 5 minutes for an entire week, which means that in contrast to the 10 individuals from the 1970s, each with 6 different measurements of glucose, here we had 1,000 individuals, each with 2,500 glucose measurements, giving us a very rich data set. But the smartest thing that happened was performed after the week of follow-up of these volunteers by a group of computational students at the lab, which use machine learning and AI technologies in order to form predictors that are aimed to predict a person's response to any given food, even foods that they've never eaten during the week of follow-up. And to make a long story short, this is one slide summarizing 7 years of work. When we calculated the predictability of the gold standard recommendations that we get when we go to a dietitian or a physician, when we want to lower our blood sugar levels, we usually get a recommendation to reduce the amount of carbohydrates in diet. There is some sense to it, and the predictability is 0.38. However, when we integrate the person-specific diet coming from the microbiome and from other measurements and try to predict the diet, the predictability is much higher. It was 0.68. And then routine drill in these kind of machine learning exercises is to freeze the predictive algorithm at some point and to apply it on a freshly blinded new cohort that was not part of the training. And when we did this on another 100 individuals on the right, you can see that the predictability remained very significant. This made us very happy, but it was now time to put ourselves to the real life test. So we recruited a number of individuals that were not included before. This time they were pre-diabetic individuals. Of all the different features of the metabolic syndrome, pre-diabetes is perhaps the most alarming feature because 40% of the adult population in developed countries suffer from pre-diabetes. Many times they don't know it. And they have a 70% chance of developing full-blown type 2 diabetes within the next decade of their life, and we have very little to offer them to reverse this trend. So we took these pre-diabetic individuals, we put them through our process, but this time we asked the machine learning algorithm to provide a set of individually good or bad diets for each individual. And of course, the diets were different from people to people because they were personalized. And we asked that the good and bad diets per given individual would be isocaloric, so we wouldn't blame the calories. And then we asked each of the participants to consume their individualized good diets for a week, then their individualized bad diets for a week, while we extensively measured them throughout the 2-week period. And to make a long story short, on the right, on the left lower panel, you can see that all the individuals that consumed their individualized good diet improved their diabetic control very drastically as compared to the bad diet. And these diets were completely counterintuitive. A person could have a good diet consisting of alcohol or chocolate, while a bad diet could in some cases be composed of tomatoes, so without measuring, you could not figure out what is good and what is bad at the individualized level. And when we revisited these algorithms after the experiment was over, we were happy to find that many of the important features that it unbiasedly used came from the microbiome. In fact, today, given the huge amount of data set that we already have, we can only use the microbiome plus a limited set of clinical features to get at a similar level of predictability. And so what we've continued to do in the last 3 years is to follow up maybe with a billion-dollar question, which is to form a double-blind clinical trial consisting, again, of pre-diabetic individuals that were randomized to the gold standard American Diabetes Association recommended diet or to the algorithm diet. And in this case, we followed them for a year to see whether we could impact real clinical features of the progress of these individuals into full-scale diabetes. And I'll just show you, the take-home message from this trial, which includes the fact that the gold standard diet does have an effect. You can see in red in 3 different readouts related to diabetic or to glucose control that it mildly improved the control in these pre-diabetic individuals. This usually is what you see in other trials, but when we use the algorithm, data-driven, individualized diet in green, the effect was dramatically improved and much more sustainable. So this was my example of how we utilize nutrition and the microbiome in order to personalize a dietary approach, in this case in diabetes, but of course we're trying to do this in many other diseases. The second avenue of data-driven microbiome treatment relates to the transfer of either individual microbes or whole microbe configurations that I've mentioned FMT, which is effective, very effective actually, in some bacterial diseases that are impacting many thousands of hospitalized patients. We've recently contributed another variant of this transplantation method by using, for the first time, transplantation of the vaginal microbiome, which is one of these non-gut microbiomes, which are much less extensively studied, but nonetheless extremely interesting. The vaginal microbiome is a simpler microbiome that is dominated by lactobacilli under normal circumstances, but a very common disease called bacterial vaginosis in women involves the replacement of the normal ecosystem with a mainly anaerobic population of microbes that often results with lots of discomfort, complaints, and lack and loss of quality of life. And you can see how this unstable anaerobic population in bacterial vaginosis looks on the right panel. What was more surprising and really alarming for us was the fact that a huge amount of the population of females suffer from bacterial vaginosis. It is estimated that 1/3 to 1/2 of all women worldwide suffer from at least one episode of bacterial vaginosis, but there is a small but actually large minority of these individuals which suffer from a particularly severe form of bacterial vaginosis, which is called recurrent or intractable bacterial vaginosis, which in contrast to the regular bacterial vaginosis is completely resistant to antibiotic treatment. And this is believed to affect 5 to 10% of the entire female population in the world. So just figure out the numbers. And in addition to low body esteem, relationship problems, and adverse effects related to antibiotic administration for long periods of time, this form of severe disease is also associated with a dramatic risk of developing a number of obstetric and gynecological complications, such as the ones listed here. So this is a huge understudied and unmet problem. So we thought that maybe we could kind of adapt the fecal microbiome transplantation in bacterial infection of the gut, which has proven to be so effective in this hugely abundant but poorly studied cureless disorder. So for a few years, we've developed a method called VMT, or vaginal microbiome transplantation, enabling the transfer of a healthy microbiome from women that are completely healthy to women that suffer from intractable bacterial vaginosis. And again, to make a very long story short, we've completed and published the first group of pilot patients suffering of the most severe intractable bacterial vaginosis that you can imagine. You can see here in the y-axis the set of clinical and laboratory criteria that quantify this disease. It's called the Amstel criteria. And you can see that all of these females suffer from the more severe form of these criteria, with spikes occurring basically every week and completely being resistant to antibiotics. And then we transplanted, using the new method, a healthy microbiome from donors into these intractable BV patients and followed them for now over 3 years. And all of these 5 patients featured a complete resolution of disease and a complete remission, which is long lasting after decades of being unresponsive to any treatment. And what was really interesting was what we found when we extensively sequenced their vaginal microbiome. You can see here one example of a principal component analysis graph in which you can see the 3 healthy donors in full colorful circles on the left side of the graph, and the 5 intractable BV patients at their starting point in the red circles on the right side of the graph. And you can see that they start from very distinct localizations, meaning that their vaginal microbiomes are extremely different than one another. But what was really interesting was what happened once we transplanted the healthy microbiome into these patients. And you can see that the configuration of the microbiome rapidly changed from their disease location towards that of the healthy donors. And it stayed there and was indistinguishable from the healthy microbiome of the healthy donors. And what we're doing now is following up on this pilot experiment with much larger experiments, controlled, randomized, involving several nations, in trying to learn more about donor compatibility and about the effectiveness of this new approach. So this was my example of how we can transplant microbes in improving the ecosystem, and through the ecosystem improve disease. The third avenue is an avenue in which we bypass the entire microbial diversity in different people and different conditions and focus on the small molecules that they secrete, these metabolites that I've mentioned before. And by understanding metabolites that are missing or overly abundant in disease, we can intervene through the metabolites while bypassing the entire inter-individual variability of the microbes themselves, which makes our life so complicated. We call this postbiotic therapy. One example of this approach, which makes me very personally excited, relates to work that we've done with a very devastating neurodegenerative disease called ALS, amyotrophic lateral sclerosis. And we chose this disease because we wanted to explore, for the first time in our case, the gut-brain axis to see whether it's for real or not. And because ALS is a disease which is cureless, it's devastating, and it's also very variable between patients. Some patients develop it at an early age, some people develop it at a late age, some patients deteriorate to full paralysis and unfortunately death within a year or two. Others take much longer to deteriorate. So whenever we see this very distinct variability in clinical traits, we think about the microbiome as a potential contributor to this variability. So we started exploring this in mice that feature a mutated gene, which is the most common form of ALS familial ALS in humans. It's called SOD1, and these are SOD1 transgenic mice. And to test for the potential contribution of the microbiome, we've given these mice broad spectrum antibiotics. And when we did this, we were surprised to find very reproducibly that upon the depletion of their microbiome, their disease became much more severe. And we measured the severity of ALS in these mice by 3 different unrelated approaches, basically testing their motor strength. So the lower the rotarod and the grip test scores in a given mouse, the weaker the mouse is, the more sick the mouse is. And the neurological score is a reverse score in which the higher the score, the more sick the mouse. And you can see that antibiotic administered SOD1 transgenic mice in red develop a much more severe and deteriorating disease in all 3 parameters as compared to the same mice having an intact microbiome. This led to a very extensive analysis of the microbiome, which is, I'm not showing you for the sake of time, and we found several microbes that help the disease deteriorate more rapidly, and one strain in our facility called Akkermansia municipula, which correlated with a inversely correlated with a more severe disease course. So the more disease became advanced, the less of this bacteria was there, suggesting that maybe this bacteria was actually improving disease. course, this was a nice observation, but since we really pride ourselves on trying to go beyond correlations and associations into causality, the next set of experiments involved the replenishment of this strain of bacteria into antibiotic-treated or germ-free SOD1 transgenic mice. And indeed, we saw that once we replenished this missing microbe, in this case, the groups with the microbe, with Akkermansia, are the black groups, disease in all three parameters became less severe or improved as compared to mice that were given either the dead bacteria or non-relevant bacteria in some other experiments. So it seems that this particular strain in our facility was improving disease course, which of course led to the next question, which is how is this mediated? So we performed a very extensive metabolomic and biochemical analysis of these strains of bacteria, which yielded several small molecule candidates that are generated by these bacteria and may mediate the beneficial effect. And for reasons that are beyond the time that we have today, we focused as one example on a molecule called nicotinamide, and because it really nicely correlated in many different aspects with the improvement that we've noted. But of course, to prove causality, we took these mice or their litimerate controls, and we replenished now the nicotinamide molecule, which was missing when the course of ALS became more severe, into these ALS-prone mice. And just like I've shown you with the bacteria, when we replenished these mice with nicotinamide, disease became dramatically less severe. This is in green this time. And survival was really substantially improved upon the replenishment with this molecule. and of course, we went to a greater extent in understanding how this molecule was being generated by Akkermansia and what it was doing in the brain. But what I want to show you is a proof of concept, a human trial, which we supplemented these mechanistic mouse studies, in which we took a group of ALS patients in Israel, and as controls, we used their healthy household members, which is it's a very stringent control for microbiome studies because people that share a household tend to share their microbiome because of common diet and so on and so forth. And despite this commonality, we found that by shotgun metagenomic sequencing of the microbiome, these ALS patients had a significantly different microbiome as compared to their household member controls. And we've also performed non-targeted metabolomics, looking at the tens of thousands of small molecules associated with the microbiome. And we found in their serum a very different signature in ALS patients as compared to healthy controls, including the nicotinamide molecule that we've seen in mice to be depleted in ALS, which was also depleted here. And we've even collected cerebrospinal fluid samples from some of these ALS patients and their controls, and we were able to show that nicotinamide was significantly reduced in its levels in the brain in ALS patients. And we're following up on these studies with more studies, trying to understand whether these molecules or any other molecules could be utilized as possible therapeutics in modulating this cureless disease. The fourth avenue that I wanted to share with you relates to a huge unmet problem. Now, imagine that you find a bacteria that is associated with disease, a bacteria that is overabundant in the microbiome. What do you do with it? And if you think about it, we really today have no means of getting rid of a bacteria when it is strongly contributing to disease. Antibiotics are a very bad way to do this. Most of these bacteria are antibiotic resistant, and you cannot give antibiotic forever. They're associated with the emergence of resistant strains, with the adverse effect. So we really have no ways of doing this. So people are trying to develop different means of eliminating bacteria in a silver bullet manner. Our approach involves bacteriophages, which are these very interesting viruses, which in contrast to the virus which we all are afraid of today, is a virus that only infects bacteria, and we consider it a good virus because the idea is that if you would take a cocktail of phages targeting a bacteria of interest through different mechanisms, you could induce a cocktail that would eliminate this bacteria in a very specific manner without greatly impacting the surrounding ecosystem, which is so important to our health. Again, I'm just showing you an example of what we've been doing for several years now. And we've basically developed this factory within the lab in which we screened and sequenced and characterized tens of thousands of phages in trying to come up with effective cocktails utilizing different mechanisms of bacterial attack and applying them into preclinical and now clinical disease. Just to show you one example, which is Klebsiella pneumonia, which is one of these antibiotic-resistant bacteria, which are implicated in a number of important human diseases. We've developed on the left side an early generation of a cocktail, which was so effective. It reduced bacterial load by, in mice by a log or two. But then we improved our use a 5-phase cocktail utilizing 5 different methods of bacterial infection. And you can see that we can completely get rid of the bacteria. And what's really cool about this method is that this is a live treatment. So if you would give a phage, as long as there is a target for this phage, the phage would propagate and would multiply within the target bacteria. So you can see here, for example, that when we give this phage cocktail once or 3 times, the impact is virtually indistinguishable because the phage is able to replicate itself as long as the target is in place. And we're now trying to move into clinical trial mode. And the last avenue that I'm going to share with you relates to the host. And we need to never forget that in these interactions, the host has a great importance, and probably we would be discovering many therapeutic targets in these communication channels that are coming from the host. So just to give you an example of a very recent work that we actually published last week, that was headed by a super talented postdoc in my lab, Alexandra Kolodzikic, that we name Ola, who was trained at a rival institute not far from where you are. And she wanted to study in her postdoc whether the host and the microbiome cooperate in modulating a very important disease called acute liver failure. And our interest in the liver spans from many different aspects. I'm also a hepatologist in my previous life, but we need to remember that the liver is the first pass organ, which sees all the metabolites and molecules coming from the gut, and therefore the liver is expected to be greatly affected by microbiome changes. And acute liver failure is one of these devastating fulminant diseases which are associated with a very high degree of mortality, 80% mortality, unless an urgent liver transplantation is allocated. And this is a disease impacting both adults and children. There are many different disorders that could lead to acute liver failure. Drug-induced hepatitis is among the leading causes, including acetaminophen or Tylenol intoxication in both children and adults. So what Ola did, she started by utilizing 2 models of acute liver failure, one by acetaminophen intoxication, another by a different mechanism, more immune-mediated mechanism induced by thioacetamide. And she combined, for the first time, to my knowledge, 2 really sexy and important technologies. One is single-cell transcriptomic, and the other relates to microbiome research in trying to understand in the best possible resolution whether we can understand this cureless and mysterious disease. And to make a long story short, Ola sequenced over 50,000 individual liver cells, and she was surprised to find 3 new cell types that have never been described before and only appear during acute liver failure. These were subsets of endothelial cells, Kupffer cells, and stellate cells. And when she looked deeply into the genetic programs of these new activated cell types, Ola was surprised to find that there is an 82 gene signature that is common to all of these 3 cells. And when she looked at these genes practically all were seemingly regulated by MYC. And you can see on the left, the promoter analysis, which basically showed us MYC in each and every one of these 82 common genes. And this could not be a coincidence. We performed many permutation analyses on the right side, and this was super statistically significant and could not be merely a coincidence, which is not biologically meaningful. Of course, this was an interesting observation, but to prove causality, what Ola did next is to apply a well-known MYC inhibitor into these mice in order to see whether the inhibition of this common MYC-regulated pathway in these 3 new cell types was changing the outcome of acute liver failure. And this was indeed the case. And she measured this using the levels of enzymes coming from the liver, the infiltration of monocytes into the liver, and of course, histology. And in all of these measures, the inhibition of mixed signaling completely reversed disease deterioration and improved mortality. Now, we, of course, performed single-cell transcriptomics in all of these interventions. And you can see on the top, the 82 genes that I've spoken before, which are the core common denominator signature of expressed genes in these 3 new cell types. On the bottom, you can see an example of one of these hallmark genes, CCL2, and you can see that under healthy circumstances, these genes are completely non-expressed. So this entire signature is off. Once you induce acute liver failure, you can see that this 82 gene signature pops up and dramatically overexpresses, including CCL2. And now when you inhibit MYC, this 82 gene signature completely disappears in all 3 cell types. So it seems that the MYC program is regulating the generation of these 3 new cell types, and through these cell types, which massively secrete chemokines and cytokines, this MYC program contributes to the vicious cycle that culminates in acute liver failure. Now, the next question that Ola asked is, does the microbiome contribute to this new mechanism? So in order to do so,
Nada, me voy a ir a casa y voy a ver si por el camino digo después me va y porque tengo que pasar unos archivos a plan y aquí no va nada y me he ido al dejo aquí.
Is this?
A mí me ha gustado, pero
He puesto una incidencia.
Could
no se lo ha gustado.
Yeah, I'll continue. So, so in order to test whether the microbiome is indeed involved in this new and interesting regulatory pathway, Ola repeated these analyses using microbiome-depleted conditions by giving broad-spectrum antibiotics or by using germ-free mice that don't have any microbiome of their own. And she performed both induced acute liver failure and performed single-cell transcriptomic analysis in these models. And indeed, she found that in the absence or in a depleted state of the microbiome, disease was much attenuated as compared to fully colonized conditions. You can see the enzymes are not jumping to the roof. You can see the cells not infiltrating the liver, and the histology was much improved. And then, of course, she looked at the 82 gene signature in the 3 cell types. I can show you here an example of 2 of the 3 cell types. And when she looked at the expression profiles, and on the y-axis, you can see the expression profile in fully colonized mice as compared to germ-free expression in the x-axis. These are mice that do not have any microbiome. She saw a highly significant increase in the mixed signature expression under colonized conditions as compared to germ-free conditions, meaning or suggesting that the microbiome was contributing to the activation of the mixed signature, leading to the development of these 3 cell types. And of course, the next question that Ola asked is, what is the mechanism by which the microbiome modulates MYC during acute liver failure? The obvious suggested mechanism would involve the secretion of small molecular patterns by the microbiome called MAMPs, or additionally by similar small molecules coming from damaged hepatocytes, which would trigger a very well-known pathway starting from toll-like receptor signaling through a series of adapters culminating in MAP kinase phosphorylation leading to MYC activation. And in order to demonstrate whether this is indeed the pathway by which the microbiome contributes to MYC activation, Ola performed the same set of experiments in mice that are deficient in toll-like receptor signaling. These are mediated TRIF double knockout mice. And she performed a series of laborious experiments in which she used pharmacological inhibitors of each of the major adapters along this very well-known pathway. You can see here an example from the mediated TRIF experiment. You can see in wild-type mice that upon the induction of acute liver failure, again, this 82 gene signature, this MYC-dependent signature, jumps to the roof. You can see all of these genes hyper-expressing, including CCL2 on the bottom. And when you do the same exact experiment in the group of mediated TRIF double knockout mice that don't have a toll-like receptor signaling, this 82 gene signature is completely attenuated, involving TLR in this pathway. And given the sake of time, I can just tell you that when she performed the pharmacological inhibition experiments, she found the exact pathway that modulates MYC signaling that is contributed by both the microbiome and the damaged hepatocytes through these small molecules. And finally, she wanted to see whether the findings in mice have any human relevance. And to this aim, we collaborated with a colleague from a tertiary center here in Israel, which performs the vast majority of urgent acute liver transplantation in acute liver failure. We allocated, with his help, sections of livers from these ALF patients that underwent liver transplantation, and section of the healthy donors that generously provided these liver donations. These are cadaveric donors. But what was important was that when Ola stained for total MYC or for phospho-MYC, she saw a very clear picture, a black and white picture in which MYC signaling was very much upregulated in the acute liver failure sections, while it was completely absent in the healthy donated liver section. And we're following up on this in trying to better understand the human situation, and maybe in the future to also intervene through one of these newly discovered checkpoint inhibitors in order to maybe provide some new forms of treatment for this cureless disease. So in this story, what Ola discovered was that under healthy circumstances, this pathway that exists in 3 resident cell types in the liver is completely shut off. So it's completely non-relevant under healthy conditions. But once you induce acute liver failure, this MYC regulating pathway turns on by factors that come from the microbiome and from the damaged hepatocytes, leading to MYC activation, which leads to the generation of the 3 new resident cell types, which cause a vicious cycle culminating in the full-blown development of acute liver failure. And what was really important for us is that a combination of microbiome inhibition and one of several distinct checkpoint inhibition that we've tested, at least in mice, resulted in a near total reversal of this process, thereby improving ALF, at least in mice. And to summarize my entire talk, what I've tried to sell to you or to convince you is that the huge amount of person-specific data that is stored within the microbiome and coupled with other non-microbiome data sets coming from humans is increasingly developed through the methods I've shown you by us, but also probably other methods that are being developed by others, into comprehensive precision medical approaches that I hope would be integrated into the personalized medicine scheme in the next decade in different combinations. And I will finish by thanking the many people at my lab coming from many places around the world to join me in this exciting, I think, journey. The very generous funding agencies that are making our work possible, our many collaborators. And as you've heard at the beginning, in the last year, we've established this cancer microbiome division with a fantastic group of people in Heidelberg, applying the same technologies and thought processes in trying to figure out whether and how the microbiome may impact cancer-related disorders. And with that, I think I'll stop here and I'll be happy to try and answer some questions.
Thank you, Eran. This was really, truly amazing, overwhelming. I'm blown away. And also, we have about 100 people in the audience who are also blown away. And there's a number of questions on the chat that I will scroll through. The first question that I see here on the chat is from Ellen Williams. And she asks, what is the role of the appendix in influencing microbiome communities?
It's a great question. And the answer is truly, we don't know. So there have been some claims over the last 2 decades that the microbiome in the appendix, in the human appendix, has a distinct configuration and distinct capacities of activating immune arms such as Th17. I'm not sure that the level of evidence is compelling enough. We know that we can live without an appendix, so I'm not sure that this is critical to our life. There have been just ran into some quite old papers suggesting that there is an inverse relationship between appendectomy and the risk of developing IBD. I'm not sure if, if this has been developed further and whether this is true or has a microbiome component. The jury is still out there.
Thank you. So I'll move on to the next question. This is a question from Toby Bonvoisin. What do we know about the microbiome in childhood and adulthood? What do we know how the microbiome in childhood and adulthood shapes immune responses to novel pathogenic viral infections like COVID?
Yeah, it's a great question. So I will start by saying that in humans, we're all born sterile, and there have been some claims that the uterus has a microbiome. I think these were probably refuted. So we're born sterile, but we acquire our microbiome from our parents and from our immediate environment. And in humans, we get the development of the microbiome completes towards an adult configuration by the age of 2 to 3. So the critical period of the shaping of a microbiome is within these 3 first years. And it is then when there is increasing evidence that suggests that perturbation of the microbiome may have long-term impacts on health later on in life, such as the propensity for obesity, asthma, and other diseases. This has mainly been shown in mice. With humans, it's more, it's more observational, but I think nonetheless potentially important and interesting. With respect to viral infection, actually yesterday there was a paper published in Cell by Dennis Kasper group showing that the microbiome through very distinct transcriptomic regulatory mechanisms impacts interferon gamma responsive genes in a way that impacts the susceptibility to develop viral infection. I don't think that they've focused on COVID. This was a mouse-based study, but it brings the very interesting prospect, which we also see in other models, that the microbiome, the bacterial microbiome, may impact more than just the bacterial kingdom. And we must not forget that there is also a virome, a very extensive viral microbiome, which is much less studied. We have a very big problem there with reference database, but there are many potentially important viruses within us that are still to be explored.
Great. Let's go on to the next question. This is a question from Simon Ledham. And the question is as follows. The results of the algorithm diet trial in the pre-diabetes are very striking. What input data do patients need to give? Do they need full microbiome sequencing? If so, can this be contracted to a panel of key bugs to improve costs?
Right, it's a great question. And the answer is that when we've started with this idea, 7 years ago, we didn't know what would be important and what would not be important. So we went to a great deal of effort and spent a lot of money in basically measuring everything that we could, because the machine learning algorithms is unbiased. It doesn't care what I want. It does its thing. So we've used all of this huge amount of data, and we finally trained it to give us a very compelling predictability. And then in retrospect, we were able to revisit the algorithm and to find that the microbiome was a big chunk, not the only chunk, but a big chunk of the readouts that it needed. So now that we have all of this huge database, we can only use the microbiome plus a very limited set of clinical features to get at a very similar predictability to the one that we've used at the beginning, without having to torture the patients with all the readouts that we originally performed.
Okay, I think Simon is very happy with that answer. Next question is from Ellen Williams. How large a part do you think microbial transplantation experiments have in mitigating antibiotic-resistant crises?
Well, it's a fantastic question, because we have a work in revision exactly on this topic, and actually I would without auto-scooping myself, I would say that the microbiome in some cases can also be considered as a reservoir of antimicrobial resistant genes and very important genes, and through through mobile elements and through horizontal transfers, these microbial resistance genes could be transferred and propagated. And the more interventions we see with the microbiome, including probiotics, including antibiotics, including transplantations, the more of these mobile elements we encounter. So this is a huge prospect that we don't really understand and could have a lot of impact on public health. We're trying to contribute a little bit of knowledge and technology in order to answer some of these questions. I hope that this will be coming out very soon, but there's vast amount of unknowns here. But for sure, the microbiome has a lot of antimicrobial repertoires that are clinically, at least potentially clinically important.
All right. We have another question from Jason Torres. Fascinating work, he says. Regarding the personalized diet algorithm study, which model features were most important in determining an individual's recommended diet? What were the particular food dimensions by which algorithm prescribed diets differed the most across individuals? For instance,
it's a great question that's very hard to answer because with machine learning, you know, whatever you put in, whatever input features you put in first, they will have the biggest weight. If you put them last, they will have less weight. So it's really interchangeable. But in a general sense, I would say that the amount of carbohydrates in diet is still a significant contributor, you know, to the predictability. It's a small, it's small in terms of the percentage contribution, but it's still very significant. But on top of it, we find microbiome features and clinical features that are easily obtainable, including previous hemoglobin A1C, diets that, or your dieting patterns the night before, and many things that we really cannot understand. The nice thing about machine learning is that you put in a lot of features into this unbiased system, and it uses whatever it wants, but then many of them, I don't understand the mechanism. It would take us decades to understand why they're important and why they contribute to the predictability, but at present we don't know, but this system allows us to bypass all of these features in order to get a clinically meaningful recommendation. But you know, the dimensionality and many of these questions are not fully understood to date by us and by other corroborative studies, including one from the UK by Tim Spector that basically reproduced our results with other machine learning technologies. And in many cases, the features are common. Not all of them are common, but the predictability is common to all of these studies.
All right. Another question from Annika Knuppel. Great to hear about this research, and especially research into BV, which is so understudied. Considering that currently the only treatment offered is antibiotics, did you observe any adverse effects in your study? Many thanks.
Yes, so you know, with this study, since this was a pilot study, we were quite apprehensive not to do any damage. So we went to a great extent, for example, in sequencing and characterizing the donor microbiome to the best possible depth in order to minimize the chances of transferring something bad into these recipients. Luckily, in these 5 cases, we saw no adverse effects, and we have been following them for 3 years, and they're all in remission. You know, one effect that may be regarded as an adverse effect is the fact that in 2 of these cases, the original transplantation was not effective, and we had to switch donor in order to make it effective. So just like in fecal microbiome transplantation, there seems to be a donor difference, a difference in the quality of donors, which, like in FMT, we do not understand. And this is why we need to greatly enlarge our population size or study size, which we're doing at the moment, in order to study more on which donors are better than others and whether indeed there are adverse effects, which we haven't run into so far.
We have another question from Toby Bonvazin. Why did data point D with intractable BV relapse? Did the anaerobic bacteria recur? And then there's another question, related question. Was the vaginal microbiome transplant a single dose, and could repeated doses be more effective?
So none of the patients relapsed. The small increase in the AMSL criteria that you're noting is because one of the 5 participants had a borderline microscopic appearance, which is one of the several features that together form these AMSL criteria, which is composed of many different clinical scores. So it kind of rose a little bit, but that patient remains to date clinically asymptomatic. So, you know, you can regard her as somewhere between partial to complete remission, depending on the weight that you give to the microscopic appearance. What was the second part of the question?
Let me reread it for you. A related question: was the vaginal microbiome transplant a single dose, and could repeated doses be more effective? Oh no, hold on.
Yes.
This is a related question. Yeah, that was the related question.
Right. So basically, all the technical details are in the published paper, but the answer is that that in the 3 complete remission cases that got a complete remission after getting their donations from a single donor, 1 transplantation was enough, and within a week you could see a clinical improvement starting to be evident. The other 2 that did not respond to the first donor necessitated a second or third transplantation from a different donor and then went into remission. So, you know, this is a too small study to really, you know, nail the protocol in terms of the number of procedures needed, but it seems to be very effective when the donor and recipient are compatible for whatever reason.
Interesting. I always wondered how you make a matrix of donors versus recipients I always wondered what would be the criteria for choosing donors for a microbiota?
Well, I can tell you that, you know, in FMT, you know, in VMT, we've performed the first 5 cases. In FMT, there are tens of thousands of cases of procedures performed. And we know for sure from a large amount of data that there is donor variability. There are some donors that are even called super donors because they seem to be the best in inducing remission, but we still have 0 capacity to identify them before we choose which donor to use. So it seems to be much more complicated than just, you know, sequencing and deciding.
So we have 2 other questions. So this is a question from Tanya Gobulichnik. Thank you for such an interesting talk. Do you observe resistance arising to phage infection over time in the resident microflora?
Yeah, it's a great question. And I would start by saying that if you put a single phage on a bacterial colony, whatever colony you choose, per definition, you would have 100% resistance developing. And the reason is that within a colony of bacteria, there are usually around 1 million copies of the bacteria. And so you'd expect to find spontaneous mutations in each of the bacterial proteins within this colony. So all you need is 1 bacteria in this colony that has a mutated receptor for this given phage, and this bacteria would eventually take over and phage resistance would develop. And this is why we took it into a great extent of generating the cocktails of phages against a given bacteria, but also making sure that each of these bacterial components or each of these phage components attacks the bacteria through a different mechanism or different receptors. So you can regard this as a cocktail similar to, you know, HIV medications. And once you have 3 or 5 phages in a cocktail attacking the bacteria through different mechanisms, the chances of resistance developing are slim, and we haven't ran under this protocol into resistance yet. All
right. So we have a related question from Simon Lidam, and he says, analogous to releasing cane toads in Australia, are there any potential issues with releasing bacteriophages in the gut, even though they don't infect host cells? I think you just mentioned.
Yeah, so first of all, yeah, there are no known eukaryotic receptors to phages. So, and phages have been used on a peculiar cultural basis in the former Soviet Union for a century as a kind of people's remedy in the absence of antibiotics. So there's lots of Russian literature, not scientifically very rigorous, but quite rigorous in terms of safety. So it seems to be a very safe approach. There are some reports non-specific, genetic material activation of toll-like receptors and so on and so forth. In our hands, they seem to be very minor. In terms of release of phages to nature, you know, in our case, we bring the phages from nature, and phages are the most common living creature, if you call them living in nature. So they're more phages than anything else around us. So I think unless you engineer them, and in some cases we do engineer them, because in some cases you cannot find a good phage against a bacteria, so you can kind of mix and match the recognition sequences within a phage to make it recognize a different bacteria. But this is the minority of cases. In most cases, we sequence and characterize natural occurring phages.
We have another question from Toby Bonvazin. What do we know about the clinical relevance of the respiratory microbiome?
Yeah, a great question. We know too little. We know using not very sensitive methodologies that the upper respiratory tract features a unique microbiome. We don't really know what it is there for, but I would guess that like any other niche in the body, it probably contributes to functions in that niche. Using much more sensitive methodologies, people are starting to also show a low biomass microbiome in the lower respiratory tract. And what's really interesting is that this microbiome may be different in disease context as compared to healthy controls. The jury is really out there. We are also trying to scrape the surface on that peculiar microbiome. But again, there's much more unknown than known. This is a 1 decade old baby field. So, you know, we would need some more time, I think, to answer these very important questions.
I agree. Time matters. And in the interest of time, the organizers are telling me, blinking me, that perhaps we should stop. It's past 6 o'clock. And I think the time we had reserved for this talk has passed by. Eran, it was really great to see you again. And your work is really magnificent and extensive, and I really appreciate that you took the time to talk about your work in this seminar series. And I think now it's time to say goodbye. And people are saying goodbye, incredible talk in the chat. And of course, I subscribe to the opinions of the audience.
Thank you, Anna. It's been my great pleasure to talk to you guys. Stay healthy and bye-bye.
Bye-bye. Bye.